While a lot of the agar that we use in the microbiology lab is used in petri dishes, we can also use agar to make solid media in test tubes. If we're doing spread plates or pour plates, or if we're making quadrant streak plates, we need a lot of surface area, so it makes sense that we would want to spread the agar out into a petri dish. But other times we're simply trying to grow. We're simply trying to grow some cells so that we can do further testing. In those instances, it makes sense that we use the solid agar in a test tube instead of a petri dish. On this slide, on the left-hand side, you're looking at two kinds of agar tubes, what we call slant tubes here, and what are called deeps here. The difference in these is that the agar in a slant tube is literally allowed to solidify on a slant. We pour the liquid agar into the tube and then we allow the tube to lean over at an angle as the agar solidifies. And you create this. You create agar that is solid on a slant. We then use the surface, that oval-shaped surface, as our growth area. We spread cells out onto the slant and we allow them to grow. Again, you would not be able to count colonies on a slant. You would not be able to do a quadrant streak and evaluate colony morphology. But if you just wanted to grow this bacteria, if you just wanted to get more cells, a slant would work perfectly fine. Now, there are other times that we create these different agar tubes called deeps. When we're making deep agar tubes, we don't tilt the tube as the agar solidifies. So the agar solidifies just as it sits in the tube. Instead of having a nice slanted surface to grow cells on, we barely have any surface area at all on the top of this agar. But that's okay, because we don't use deeps to grow surface cells. Instead, we poke the cells down into the agar using a wire. The reason you would do that is if you were working with a bacterium that was sensitive to oxygen and needed to be in an oxygen-free or at least an oxygen-reduced environment. We take a tool called an inoculating wire. In other words, it doesn't have a loop on the end, it's just a sharp wire. And you dip it into your sample or your culture, and then you poke the wire down into the agar. You'll be depositing cells along that line. And if you put this tube into the incubator, those cells will grow. In fact, they'll grow out into the agar sometimes, depending on the species. So, slant tubes are solid agar in a test tube that are allowed to dry on a diagonal. That increases the surface area to allow us to grow some cells. We're not going to use slant tubes to count colonies. We're not going to use them to evaluate colony morphology. We're just trying to get some cells to grow. Deeps are a little different. Deeps allow the agar in the test tube to simply harden in place, providing nice deep areas where we can inoculate cells into an oxygen-reduced area. We use an inoculating wire here to introduce the cells deep down into the agar. In comparison, we would use a regular inoculating loop to inoculate the surface of a slant. The last thing we're going to be talking about today is a little more detail about colony morphology. Remember, a lot of what we do in the microbiology lab is identify microbes. We try to find out what the genus and species of the microbe is so we can report back. In our evaluation of these bacteria, we're gathering bits and pieces of information about them with different tests that we run, including growing them in different types of culture media. We can use solid media, like these agar plates, to grow bacteria in different ways. If we put lots and lots of cells on the surface of an agar plate, we're going to get this solid sort of mat of growth. We call this type of a mat a lawn. It's just a solid convergence of bacterial cells. Now, why would we ever want to grow a lawn on purpose? Well, sometimes we want to test to see whether bacterial cells are sensitive or resistant to an antibiotic. That's what you're seeing on this plate in this image. That little white circle in the center of the image is actually a disc made out of paper that has been impregnated with an antibiotic. What we do in the lab when we want to test for antibiotic sensitivity is we put lots and lots and lots of cells onto an agar plate, and then we press these antibiotic discs down on top of the cells. We put the plate into the incubator and we see what grows and what doesn't grow. Now, over here, this plate doesn't have any discs on it, and you can see that over 24 hours, the whole surface of the plate became covered with cells. Again, this is what we call a lawn of cells. But over here, we see something different. Notice that there's a zone around the antibiotic disc where there are no cells growing. Notice that there are cells growing out here, far away from the antibiotic disc. But any of the cells that were up close enough to the disc to be exposed to that antibiotic, they didn't grow. In fact, they died overnight, creating this clear zone, or what we call a zone of inhibition. This is how we test antibiotics in the laboratory, by creating these lawns of cells. Now, many other times we want to grow colonies, including the times that we're trying to evaluate colony morphology. Colony morphology, or the body form of a bacteria growing as a colony, is one of the traits that we can use to identify bacteria. In other words, each species of bacteria has its own colony morphology. When it grows as a colony, the colony will look a particular way. What kind of things do we look for? Well, we look for the size of the colony, the shape of the colony. We look at the color of the colony, whether or not it's shiny or dull on its surface. We look at the margin of the colony or the edge and evaluate whether it's smooth or rough. We look at how elevated the colony is. Is it growing up high or is it more flat down near the surface? By evaluating all of these physical traits of the colony, we can get information to help us determine, along with many other tests, what particular species of bacteria is growing. This is a chart that comes out of a microbiology textbook, and it gives you a visual representation of some of the ways that colonies grow on agar plates. Up at the top here, you can see a drawing of many different kinds of shapes of colonies and the words that we use to describe them. A lot of bacterial colonies are just circular. They grow as a circle of some diameter. Some colonies, though, grow as very small circles, and those are referred to as punctiform colonies. You don't need to memorize these terms, by the way. I'm not going to test you on any of that, but I do want you to understand that colonies grow with different sizes and different shapes. Some of them look quite unusual, like this rhizoid colony or this filamentous colony. Some of them have quite uneven shapes, irregular as they're called. Now, in the middle, you're looking at images of colonies from the side view. Notice that some colonies grow quite flat, while others grow quite raised. There are convex colonies. There are colonies that are dome-shaped. There are even colonies that look like fried eggs from the side. Those are called umbonate colonies. Colonies have different margins or edges. Some of them are smooth and even. Some of them are uneven and wavy, filamentous, lobulate, and so on. So we examine individual colonies and we record all of the features of those colonies so that we have more information about what possible organism we're dealing with. On this slide, you're looking at some actual colonies growing on agar plates. Now, if you take a look at this image on the upper left, right away you see that there are different colonies growing on this plate. Some of them are rather large. Some of them are rather small. Most of them are circular, but some of them have a yellow color and some of them have a more grayish-white color. There are several different species of bacteria growing on this plate. There's Staph aureus, that's the yellow one. E. coli, that's this one. There's Enterococcus faecalis, that's the little one. When you have different types of bacteria growing on a plate, you often see different colony morphology as well. And that's quite helpful if we're looking at an environmental sample that has multiple types of bacteria in it. It is important to know one thing, however, and be sure to write this down in your notes. While each individual species of bacteria has its own colony morphology, more than one type of bacteria can have the same colony morphology. In other words, if you see colonies on a plate that all look the same, you could have one species of bacteria growing, or you could have two species or more growing that all have the same colony morphology. So we can never look at a plate and know for sure that we have only one species growing. We have at least one species growing. Make sense? If you look at this picture up here, again, you can see multiple colonies on this plate and multiple types of colonies. I've got these larger circular colonies growing that have a nice smooth margin. But down here I have a large colony type growing that has an undulate margin or an uneven margin. See that? I've also got some kind of an organism growing that produces small colonies. I may even have another organism down here growing, producing even smaller colonies. So it is quite common for us when we're looking at environmental samples to see multiple types of colony morphology. We have to be careful, though, when we count different types of colonies on a plate. We have to be careful. We have to remember that sometimes two different species of bacteria can have identical colony morphology. Down here, this last image I wanted to show you because of its remarkable color. This is an organism called Seratia Marcescens. This is a pathogen in humans, and when you grow it in the laboratory, you get this remarkable red color. The colony morphology of Seratia Marcescens is otherwise pretty unremarkable. It grows as small 1 to 2 millimeter circular colonies, otherwise pretty boring, except for the fact that it grows with this bright, brilliant red color. When we see that red color on a plate, one of the first things we think about then is that it might be Seratia. We're going to have to do other testing, though, to confirm that. The samples that we gather from the environment often have multiple types of bacteria growing on them. And sometimes we have multiple types of organisms. Both bacteria and fungi can grow on our agar plates in the microbiology lab, and fungi also have colony morphology. You cannot tell the difference between a bacterium and a fungus on an agar plate. Even though sometimes we look at certain colonies and we think, gosh, that looks really fuzzy, and that makes me think of fungus. You cannot know that that's a fungus until you look at it under the microscope. If you see multiple different types of colony morphology, you know you have multiple types of organisms growing on that plate. It's a little difficult to see, but if you look at this colony, for example, and this one here, they have that fried egg appearance. See how the center of the colony is just a little bit more white than the rest of the colony is. We have that organism growing. We also have some organism growing as a small white colony. We also have an organism growing as a larger white colony. We've got something over here with a rhizoid shape. Lots of different kinds of bacteria growing on this plate. Over here we have yet another plate with multiple species growing on it. If you see two colonies that have different colony morphology, you know that you're looking at two different species. So for example, this larger type of colony is a different species than this smaller white colony. That I can be sure of. What I can't say is that all of the small white colonies on this plate come from the same organism, because there may be two or more organisms that grow as small white colonies. Here's a couple other plates taken from the environment. Look at the array of organisms that you can grow. Look at that amazingly large and irregular looking colony growing on this plate. Very interesting morphology there. And then we've got something that looks much more boring, just a circular colony, maybe 2 millimeters across in diameter. They vary, microbes vary terrifically in colony morphology. Over here, this person has labeled all of the different varieties of colonies growing on this plate. Look at the different sizes, the different shapes, the different colors. Remember, they sometimes have different elevations. They have all kinds of characteristics. that make colonies unique. And remember, you cannot look at a colony and know for sure that you have a bacterium or a fungus. You have to look at the cells under the microscope in order to know that. If you look on this slide, if you look at this image over here on the left, you might suspect that this is bacteria growing, different species of bacteria. I've got one colony that's quite large. It's lighter colored in the center, darker at the edges. I've got these smaller circular colonies growing. It looks like bacteria. In comparison, over here on this plate, I've got something really fuzzy growing, something quite irregular and really fungal looking. But guess what? It turned out that this organism is actually a bacterium, and this is actually a fungus. So remember, you cannot judge whether you've got a bacterium or a fungus until you take cells under the microscope. The next time we get together, we're going to be talking about the fungi. We'll spend our time together learning a few things about these fascinating organisms. And we'll see you then.